Welcome back to the Deep Dive. Today, we are diving deep into a subject that sounds, well, pretty dark on the surface, cell death. But it's actually the engine behind so much of life, health, and, disease regulation, too. Our mission today is really to get past this simple idea of cells just dying. We've got a stack of sources here, and we want to unpack the really critical difference between the two main ways cells check out. One's kind of messy and violent. The other is almost surgical. Understanding this split is kind of a shortcut to grasping a huge amount of human pathology.
Absolutely. And what's fascinating right from the start is that cell injury, it's really a spectrum. If the stress is mild, maybe brief oxygen loss, the cell can bounce back. It's reversible. You might see some swelling, what we call hydropic change. That's because the cell's energy-dependent pumps, the ATP pumps that manage water, they falter a bit.
Right.
But then there's this crucial point, this point of no return. Once a cell crosses that line, it's committed, and it heads down one of two main paths to, well, total failure, necrosis or apoptosis.
Okay, so we can frame it right there. Necrosis is the big, messy accident, like a catastrophic failure.
Exactly.
And apoptosis is more like a pre-planned, highly controlled cellular suicide mission.
A scheduled demolition, yeah. That's a good way to put it.
All right, let's start with the explosion then, necrosis. You're saying this is always pathological, always bad news.
Always pathological, yes. It's the result of really severe injury. Things like profound lack of oxygen, what we call ischemia, or overwhelming toxins, major physical trauma. The defining event in necrosis, the absolute key, is the catastrophic failure of the cell membrane. It just breaks down.
And everything leaks out.
Everything leaks out. The cell basically gets digested from the inside by its own enzymes leaking from lysosomes. But the real trouble starts when all that internal stuff spills into the neighborhood, into the surrounding tissue. Those leaked components, they have a name. DAMPs. Yep. Damage-associated molecular patterns.
DAMPs. Okay, that sounds ominous. What are these things? Normal stuff inside the cell, but suddenly toxic outside.
Precisely. Things that are essential inside the cell, like ATP from the mitochondria or uric acid from DNA breakdown, become powerful danger signals when they're detected outside. The immune system sees these DAMPs and immediately sounds the alarm. It triggers a big inflammatory response.
Ah, okay. That's why necrosis is always associated with inflammation and swelling, right?
Yeah.
It's the collateral damage from the leakage.
Exactly right. It's messy and involves the surrounding tissue.
And this leakage isn't just some, microscopic detail. It's actually how doctors figure out something terrible has happened, isn't it?
Oh, absolutely critical for diagnosis. It gives us vital biomarkers. Think about a heart attack.
Okay.
Cardiac muscle cells die by necrosis. They leak proteins specific to heart muscle, like cardiac troponin, into the bloodstream. A quick blood test shows high troponin levels, and bam, that's your diagnosis of a myocardial infarction, often way faster than imaging could show it. Necrosis literally leaves a biochemical fingerprint.
Wow. Okay, so if we were looking at this under a microscope, what does this violent death actually look like? You said messy?
Visually, it's quite distinct. Necrotic cells stain intensely eosinophilic. They turn this bright pink or red color with standard H&E stains.
By pink?
Well, they lose their RNA, which normally stains blue, and what's left are all these denatured proteins inside the cell, and they really soak up the eosin, the pink dye. Then you see the nucleus just fall apart. It happens in stages, typically.
Okay, the sources mention these three terms that always trip me up. Karyolysis, pinosis, and Injurious. How do we keep those straight?
Think of the sequence. First, the nucleus starts to fade. That's karyolysis. The chromatin kind of dissolves as the DNA breaks down.
Fading. Got it.
Then the nucleus shrinks down into this small, dense, dark blob. That's pinosis. Think P for packed. Condensation.
Shrinking in dark. Okay.
And finally, that shrunken nucleus fragments, it shatters into little pieces. That's karyorexis, like an explosion or wreck.
Fading, shrinking, shattering. Okay, that helps.
Karyolysis, pinosis, karyorexis.
And the sources emphasize that how this looks in the tissue, the overall pattern, can tell you a lot about what caused it.
Oh, definitely. These patterns are key for pathologists. The most common one is coagulative necrosis.
Coagulative.
Yeah, you see this typically in infarcts, like in the heart or kidney, basically anywhere except the brain when it's due to lack of oxygen. And what's really striking is the tissue keeps its shape. It feels firm. The basic architecture is preserved, sometimes for days.
If the cells are dead.
Because the injury is so severe, it denatures everything, not just the structural proteins, but also the digestive enzymes that would normally break the tissue down. So the structure remains like a ghost outline.
Dead but standing firm. Okay.
Exactly. Now, the complete opposite is liquefactive necrosis. Here, the tissue literally turns into a viscous liquid. Ooh.
Liquefies. Okay, where do we see that?
Classic example is in bacterial infections. Think of pus in an abscess. That's basically liquefied tissue mixed with dead white blood cells thanks to all the enzymes released by the bacteria and the immune cells. And interestingly, lack of oxygen, hypoxia in the brain, always causes liquefactive necrosis. The brain just melts.
Yikes. Okay, so coagulative holds its shape, liquefactive melts. What else?
Then there's caseous necrosis. Caseous means cheese-like.
Cheese-like.
Yeah, it looks like crumbly white or yellowish cottage cheese. It's really distinctive, kind of granular debris. This is the absolute hallmark of tuberculosis, TB infections.
Ah, okay. TB equals caseous necrosis. Easy to remember.
Pretty much. And finally, there's a specialized type called fat necrosis. This is specific destruction of fatty tissue.
How does that happen?
The classic scenario is acute pancreatitis. When the pancreas is inflamed, digestive enzymes, specifically lipases, leak out. These lipases break down triglycerides in fat cells, releasing fatty acids. These fatty acids then combine with calcium ions in the tissue.
Calcium, like in soap.
Exactly like soap making. It's a process called saponification. It creates these chalky, white, soap-like deposits in the dead fat tissue. You can often see them grossly.
Fascinating. So these different patterns are really diagnostic clues. Okay, so that's the messy, inflammatory explosion of necrosis. Let's switch gears to the other side. Apoptosis, the controlled demolition crew. You said the key difference is the lack of inflammation. It's quiet.
Completely quiet. Apoptosis is this highly regulated, genetically programmed pathway. It's designed to eliminate unwanted or damaged cells with surgical precision without upsetting the neighbors.
And it's not always about damage, right? It's essential for normal life.
Oh, absolutely essential. We rely on it constantly. It maintains homeostasis, keeping the right number of cells in tissues that turn over rapidly, like the lining of your gut. It's crucial during development. You know, the reason you have separate fingers and toes instead of webbed hands and feet. Apoptosis removed the cells in between.
Okay. Programmed cell death in action.
Exactly. And critically, it gets rid of potentially dangerous cells, like lymphocytes, immune cells that might react against our own tissues. Apoptosis eliminates them before they can cause autoimmune disease.
So structurally, how does it differ from necrosis? Necrosis swells and bursts.
Yeah.
Apoptosis.
Apoptosis shrinks, the cell pulls away from its neighbors, the cytoplasm gets denser. But the most characteristic feature happens in the nucleus. You get this dramatic chromatin condensation. The DNA clumps together into these dense masses right up against the nuclear membrane. It looks very different from the fading or shattering in necrosis.
Shrinking and clumping. Okay.
Then the cell breaks apart, but not in a messy explosion. It fragments neatly into these small, membrane-bound packages called apoptotic bodies.
Little tiny bags of cell debris.
Precisely. And this brings us to the molecular machinery. The real executionals of apoptosis are enzymes called caspases.
Caspases. The cellular scissors, I've heard them called.
That's a good description. They are specific types of proteases, cysteine proteases. Once they're activated, particularly the executioner caspases like caspase 3, it's game over. They chop up key proteins in the cell, structural proteins, proteins that manage DNA, leading to that fragmentation we talked about.
But how does the body clean up those apoptotic bodies without causing inflammation? If they just floated around.
That's the really elegant part. The removal, called efferocytosis. It relies on a specific eat-me signal.
An eat-me signal, seriously.
Seriously. In a healthy cell, a particular phospholipid called phosphatidylserine is kept strictly on the inner surface of the cell membrane. When apoptosis kicks in, enzymes flip this phosphatidylserine to the outer surface of the membrane, including on those apoptotic bodies.
So it's like waving a flag.
Exactly. Phagocytic cells, like macrophages patrolling the tissues, have receptors that recognize that flipped phosphatidylserine. They see the signal and gobble up the apoptotic bodies very quickly and efficiently.
Wow. So the cleanup is built into the process, preventing any leakage.
Precisely. No damps released, no inflammation. Neat and tidy.
Okay, so the caspases are the executioners. But how does the cell decide to pull the trigger? How are they activated? There are two main routes, right?
That's right. Two main pathways converge in activating those caspases. The most common one, the one used most often inside the body, is the intrinsic pathway. It's also called the mitochondrial pathway.
Intrinsic meaning? The signal comes from inside the cell.
Exactly. It's triggered by internal cellular stress, things like severe DNA damage that can't be repaired, or a buildup of misfolded proteins, or even just the loss of essential survival signals the cell needs. This internal stress disrupts a delicate balance involving a family of proteins called the BCL2 family.
The BCL2 family. Okay, this sounds important. Let's break this down. Who are the players here?
Okay, think of the mitochondrial outer membrane as a critical gate. There are proteins from the BCL2 family that are anti-apoptotic. Their job is to keep that gate closed and secure. The main ones are BCL2 itself and BCL-XL. They're like the guards protecting the cell, saying, don't die.
The pro-survival guys. Okay.
Then you have the pro-apoptotic members of the family. The key executioners here are two proteins called BX and BAK. They're the ones that, when activated by stress, want to punch holes in that mitochondrial gate.
So it's a constant battle. BCL2 and BCL-XL versus BX and BX.
It's a constant molecular tug-of-war, yes.
Yeah.
Under normal, healthy conditions, the pro-survival guys keep BX and BX in check.
But wait, why have this conflict? If the cell is damaged beyond repair, why have proteins actively fighting against dying? Seems inefficient.
But it provides a crucial checkpoint. The cell doesn't want to trigger self-destruction for every minor problem. This balance allows the cell to sense the level of stress. Is it manageable? Can we repair? Or is it truly catastrophic?
Okay, so it's a threshold mechanism.
Exactly. When the stress signals become overwhelming, the balance tips. BX and BX overcome BCL to BCL-XL. They get activated, form channels or pores in the outer mitochondrial membrane.
They punch holes in the mitochondria when it comes out.
The critical molecule that leaks out is cytochrome C. Normally, it's part of the electron transport chain inside the mitochondria involved in making energy. But when cytochrome C leaks into the cell's cytoplasm, it binds to other proteins to form a complex called the apoptosome. And the apoptosome's job is to activate the key initiator caspase of this pathway, caspase 9.
And caspase 9 then activates the executioner caspases like caspase 3.
And down goes the cell. That's the intrinsic pathway in a nutshell. Stress stench, mitochondrial leak, cytochrome C release, caspase 9 activation, execution.
Okay, clear. What's the second pathway, the extrinsic pathway?
Right, the extrinsic or death receptor pathway. As the name suggests, this one is triggered by signals from outside the cell.
Like a direct order to die.
Pretty much. It uses specific receptors on the cell surface called death receptors. The most famous one is called FAS, also known as CD95. There's a corresponding molecule called FAS ligand, or FASL. Often, immune cells, like cytotoxic T lymphocytes, will display FASL on our surface.
So the T cell finds a target, like a virus-infected cell or maybe a tumor cell.
Exactly. The T cell docks, its FASL binds to the FAS receptor on the target cell. This binding causes multiple FAS receptors to cluster together. This clustering recruits special adapter proteins inside the cell, forming a complex right under the membrane called the DISC, the death-inducing signaling complex.
DISC.
And the DISC's function is very direct. It recruits and activates another initiator caspase, caspase 8.
So extrinsic pathway directly activates caspase 8, intrinsic pathway activates caspase 9, but both end up activating the same executioner caspases.
That's right. Both caspase 8 and caspase 9 can activate executioner caspases like caspase 3, leading to the cell's demise. The extrinsic pathway is often used by the immune system to eliminate specific unwanted cells.
Okay, this distinction between necrosis, messy accident, and apoptosis, clean suicide, seems pretty clear. It held up for a long time. But the research doesn't stop right. The sources indicate things are actually, well, a bit more complicated now. The lines are blurring.
They really are. Over the last decade or two, researchers have identified several other ways cells can die, and crucially, some of these are regulated pathways like apoptosis, but they look more like necrosis. They cause inflammation and membrane rupture. It shows the cell has backup plans, maybe overlapping systems. It's opened up whole new areas for thinking about disease treatment.
Okay, let's get into these newer pathways. First up is one called necroptosis. The name itself sounds like a hybrid. Programmed necrosis.
That's exactly what it is. Morphologically, it looks like necrosis. The cell swells, the membrane breaks, it releases damps, triggers inflammation. But the key difference is that it's triggered by specific signals and driven by a defined molecular pathway. It's regulated.
Okay, but why? If the cell has the clean apoptosis pathway, why evolve a programmed explosion? What's the advantage? Seems counterintuitive.
Redundancy seems to be the key driver. Think of it as a backup self-destruct mechanism, especially important when apoptosis is blocked. Some viruses, for instance, are very good at producing proteins that inhibit caspases, trying to keep the host cell alive so the virus can replicate. Necroptosis provides a caspase-independent way for the cell to die anyway, sacrificing itself to limit the infection.
Ah, clever. A way to bypass viral sabotage. So if it doesn't use caspases, how does it work?
It's often triggered by the same sorts of receptors as the extrinsic apoptosis pathway, like TNFR1, the receptor for TNF-alpha. But instead of activating caspases, the signal activates a pair of kinases called RIPK1 and RIPK3, receptor interacting protein kinases.
IP kinases, okay.
These RIP kinases then phosphorylate another protein called MLKL, and phosphorylated MLKL is the executioner here. It moves to the plasma membrane, inserts itself, and basically punches holes in it, causing the cell to swell and burst.
Like a molecular wrecking ball activated by RIP kinases.
A good analogy. And because it causes inflammation, necroptosis is now heavily implicated in various chronic inflammatory diseases and also in neurodegenerative conditions like Parkinson's or ALS. There's a lot of interest in developing drugs that block RIPK1, for example.
Fascinating. Okay, next hybrid. Pyroptosis.
Yeah.
Sounds fiery.
It is. Pyroptosis means fiery death. It's another form of programmed cell death, but its defining feature is the release of potent pro-inflammatory cytokines, especially interleukin-1, IL-1, which is famous for causing fever. Pyro refers to fever.
So programmed death plus fever and inflammation, when does this happen?
It's primarily activated in response to microbial infections. Certain bacterial components, for example, trigger the formation of a large intracellular protein complex called the inflammasome.
The inflammasome.
Yeah. The inflammasome's main job is to activate another specific caspase, caspase-1. Now, caspase-1 is different from the apoptotic caspases like -3, number 8, -9. Caspase-1 does two things simultaneously. It processes the precursor form of IL-1 into its active fever-inducing form, and it triggers the cell to swell, rupture, and die, releasing that active IL-1 and other inflammatory signals.
So it directly links cell death to a strong inflammatory alert signal.
Exactly. It's a way for infected cells, especially immune cells like macrophages, to die while simultaneously sending out a powerful danger signal to recruit more immune help.
All right, what else is new on the cell death menu? There's one discovered relatively recently, apoptosis.
Yes, apoptosis was described formally around 2012. It's completely different mechanistically from apoptosis or necroptosis. The name gives a clue. Ferro means iron. It's an iron-dependent form of cell death.
Iron-dependent, how does that work?
It's triggered by a massive buildup of oxidative stress, specifically involving the peroxidation of lipids in the cell membranes. Think of it like the cell membrane rusting uncontrollably. This process requires iron ions to catalyze the reactions. If you have too much lipid peroxidation, the membrane integrity fails and the cell dies. Crucially, it's not dependent on caspases or RIP kinases.
So it's all about runaway membrane damage driven by iron and oxidation.
That's the core of it. And you can often prevent ferroptosis by removing excess iron or by boosting the cell's antioxidant defenses against lipid peroxidation.
And this is relevant in diseases.
Hugely relevant. It's implicated in the kind of damage you see after a stroke when blood flow returns reperfusion injury, which causes a burst of oxidative stress. Also potentially in some neurodegenerative diseases and even certain cancers. It's a very active area of research.
Okay, one more. The sources mention autophagy, but you said earlier this is more about survival.
Right. Autophagy literally means self-eating. Primarily, it's a survival mechanism. When a cell is starved or stressed, it can wrap up damaged organelles or chunks of cytoplasm in membranes, forming autophagosomes. These then fuse with lysosomes, and the contents are digested and recycled. It's the cell basically cleaning house and cannibalizing non-essential parts to generate fuel and building blocks to stay alive during tough times.
So self-eating to survive, how does it relate to cell death then?
It's a bit of a double-edged sword. In many situations, autophagy helps the cell cope with stress and prevents death. But if the stress is too severe or prolonged, the autophagic process itself can become overwhelmed or dysregulated. In some contexts, excessive or failed autophagy seems to contribute to or trigger cell death, sometimes apoptotic death, sometimes other forms. Its role is complex and context-dependent. We see this complexity in cancer, where autophagy can help tumor cells survive chemotherapy, bad for the patient, but in other situations, inducing autophagy might kill cancer cells. It's also linked genetically to inflammatory bowel diseases through genes like ATG1601. It really highlights that the line between survival and death pathways can be very thin. So if we try to wrap this all up, the big takeaway is that cell death isn't just one thing. It's not binary. It's this incredibly sophisticated, multi-layered system. Critical life or death decisions are being made constantly at the molecular level by that balance of BCL2 proteins at the mitochondria, by the activation of different caspases, by these RIP kinases.
Yeah, it's moved way beyond just necrosis is the accident and apoptosis is the plan. Necrosis is violent, inflammatory, yeah, usually accidental. Apoptosis is precise, quiet, programmed. But these newer forms, necroptosis and pyroptosis, they show us cells have these regulated, programmed ways to die messily if needed. Redundancy, backups.
Exactly. Multiple ways to ensure controlled self-destruction when things go wrong.
So thinking about this from the listener's perspective, what's the implication? We've talked about all this intense molecular regulation, BCL2, BAX, BAK, RIPK, caspases. Does manipulating these specific molecular switches, these checkpoints, represent maybe the most powerful way forward for treating diseases that currently seem intractable? Imagine if we could reliably force cancer cells into apoptosis by deliberately tipping that BCL2-BAX balance, or stop the devastating neuron loss in Alzheimer's or Parkinson's by specifically blocking RIP kinases involved in necroptosis.
That's precisely where a lot of cutting-edge research is focused, developing drugs that target these specific nodes in the death pathways.
It feels like understanding this microscopic battle for survival, life versus death at the cellular level, really does hold the key to tackling some of our biggest health challenges. Well, that's all the time we have for this deep dive. Thank you for joining us as we explore the intricate world of cell death. We'll see you next time.